Hello and welcome. In this briefing, we're doing a deep dive into the mumps virus, and we're going to focus specifically on the challenges and, the big diagnostic shifts happening right now in the Netherlands.
So let's start with the question that I think is on everyone's mind. We have a fantastic national vaccination program, so why are we still seeing these mumps outbreaks, especially among young adults? Well, the answer, as you'll see, is pretty complex. It has to do with the virus itself and, crucially, how we're testing for it.
Just a quick heads up before we jump in. All the information you're about to see was compiled using a Gemini deep search. And if you want to explore the data for yourself, the full list of sources is right there for you in the linked Notebook LM document. Okay, so first things first. Let's get a quick refresher on the fundamentals. What exactly is the mumps virus and what does it actually do to the body?
All right, so let's look at its family tree, so to speak. The mumps virus is a single-stranded negative sense RNA virus. You can see here it's part of the Paramyxoviridae family, which makes it a cousin to other viruses you're definitely familiar with, like measles and parainfluenza.
Now, take a look at this structure. The virus is what we call pleomorphic, which just means it doesn't have one single fixed shape. But the parts that really matter are its lipid envelope on the outside and that helical nucleocapsid tucked inside. And you see those little spikes? Those are absolutely key. They're what the virus uses to grab onto and infect our cells.
And this highlights a distinction that is just so important to understand. From an antigenic point of view, there's only one serotype of mumps virus. Now, this is fantastic news because it means our vaccine, which is based on genotype A, it works against all the strains that are out there. But genetically, it's a different story. There are lots of genotypes, from A to N. And while that doesn't help the virus dodge the vaccine, it gives us in public health an incredible tool to track the exact transmission chains of an outbreak.
So clinically, what do we see? Well, after a pretty long incubation of 12 to 25 days, you get that classic hallmark sign, acute parotitis. But we also have to be on the lookout for serious complications, things like aseptic meningitis or orchitis in postpubertal males. The biggest public health challenge, though, it's that incredibly high rate of asymptomatic infection. This is especially true in people who've been vaccinated, and it's what allows the virus to just spread silently through a community.
And let's be crystal clear about treatment. There is no specific antiviral therapy for mumps, not here in the Netherlands, not anywhere else. Management is 100% supportive care, rest, fluids, and managing the symptoms. And that's why we're not getting into things like antiviral resistance or pediatric dosing. Since there's no specific drug, those topics are off the table. This lack of a direct treatment is precisely why getting the diagnosis right and preventing the spread are so incredibly important.
Okay, so if we can't treat it, everything, and I mean everything, hinges on getting an accurate and fast diagnosis. And this is an area where we've seen a massive shift in the modern lab.
This quote from the source material really just says it all. The old methods we used to count on, they're just not fit for purpose anymore, especially not in our highly vaccinated population.
This table lays out the new reality perfectly. Real-time RT-PCR, done on a buccal swab that's collected within 3 days of symptoms starting, that is the undisputed gold standard now. Viral culture, it's just too slow, it's too inconsistent. But the biggest change, the one we all need to internalize, is that IgM serology is now considered highly unreliable in our population.
And this right here is the single most important diagnostic message of this entire briefing. In someone who's been vaccinated, a breakthrough mumps infection often triggers a very rapid memory IgG response. That response can completely shut down or even prevent the IgM response we would have traditionally looked for. So if you're relying on IgM serology in the Dutch context, you are going to miss cases. It's as simple as that. The priority has to be getting that early buccal swab for RT-PCR.
Okay, so if we can't treat it, and the diagnosis has to be just so, then our best line of defense, our only line of defense really, is prevention and really solid public health control.
When we talk about control, it really boils down to a few key numbers you need to remember. The first one is 5. According to Dutch public health guidelines, a patient with a confirmed case of mumps has to be isolated for 5 days after that swelling starts. That 5-day window is absolutely critical because it covers the period of peak viral shedding.
The second key number is 2. The entire Dutch Rijksvaccinatieprogramma is built around 2 doses of the BMR, or MMR vaccine. The schedule, one at 14 months and another at 9 years, is designed to build really strong immunity across the population. And, for decades, it's worked incredibly well.
And the third critical number is 1. Mumps is a Group C notifiable disease. That means clinicians and labs have just one working day to notify their local GGD, the Public Health Service. This rapid reporting is the starting gun for the entire public health response.
And that single notification from the lab, it triggers this whole vital cascade. The GGG jumps in, they verify the case, they start tracing contacts, they analyze the risk of more people getting sick, and they provide that crucial control advice. This can even mean offering vaccinations to close contacts to shore up any local gaps in immunity. It all starts with that one lab confirmation.
Which brings us full circle right back to that question we started with. The scientific consensus is clear. The mumps outbreaks we're seeing today are not being caused by some new vaccine escape variant. They're being driven by the very natural waning of vaccine-induced immunity years and years after those childhood shots.
And, well, that conclusion naturally leads us to the next big question, the one that's being debated right now in public health circles. If waning immunity is the problem, is a third MMR dose maybe offered to adolescents or young adults? Is that the future of mumps control? It's a really important strategic question to think about.
And that wraps up our deep dive into the mumps virus in the Netherlands. We really hope this has been a valuable update for you. For more expert briefings like this one, please make sure you subscribe. And we'd really value hearing from those of you on the front lines, so please share your own insights or questions in the comments below. Let's keep this conversation going.